Hello students, welcome to EPG Pathshala. I am Vidya a former professor of biophysics at All India Institute of Medical Sciences, Delhi. I was also professor of bioinformatics at JP Institute of Information Technology in Noida and professor of bioinformatics and director of Dr. D.Y. Patil Biotechnology and Bioinformatics Institute. Actually, my passion is teaching. I have been a teacher for more than 50 years. Today, we are going to discuss about a module, interactions of small molecules with proteins. This come under the paper Biomolecules and their interaction. Small molecules, water and iron all bind to proteins and control host of biochemical interactions. One of the earliest recognized examples of binding of a small molecule like O2 to hemoglobin. This initiates transport of oxygen throughout our vascular system. Small molecules can be cofactors in biochemical interactions. They can be activators or inhibitors of enzymes, a principle used for drug designing, and it has great importance in pharmaceutical industry. These participate in host of enzyme catalysis, signal transduction, immune system, and isolation of proteins. So students, what are you going to learn in this lecture? I'll first make it clear to you. Like, I want to start with the basic thermodynamic and physical basis of small molecule and protein interaction, because a small molecule binds to protein only if it likes it more than binding to solvent. Next, I want to describe experimental and theoretical techniques used, because it is first necessary to understand how can we study the interaction, and then only we can decide whether this interaction was better than other interaction. Next to it, I want to quote and describe you a couple of examples of small molecule and protein interactions. The first example which I want to give you is a very important topic, that is interaction of HIV-1 protease with inhibitors. This is used for designing of drugs against AIDS. Next, another very important topic, that all of us get pains, swellings so much. So we eat always brufen, ibuprofen and drugs which are non-steroidal anti-inflammatory drugs, or NSAIDs as these are called. What are these? These are inhibitors of cyclooxygenase 2 or COX-2, selective inhibitors. Next, I want to describe you the example of how viral peptides are recognized by class 1 MHC proteins. Again, this is very important issue in immunology. Then I can go on like that for giving you innumerable examples, but since our time is limited, I want to give only one more example, that is activation of a proton pump by interaction of a retinol with the membrane binding protein bacteriorhodopsin. Interaction of small molecule or ligand with protein. A tight complex between the ligand and protein forms only when the interaction between them is more favorable than the interaction of the ligand or protein with the solvent. We need the knowledge of the two surfaces and also balance of forces, because the first most important thing for interaction is the structural compatibility between the ligand or a small molecule and protein. Thermodynamic basis of small molecule protein interaction is very important. The interaction involves physical contact between the ligand and its receptor or protein for a certain length of time. It depends on the concentration of the ligand L and protein in salt, and the equation reaction generally proceeds as RL, forward or backward, equal to R plus L, and the equilibrium constant Kd is given as Kd equal to R into L divided by RL, the concentration of complex. Enzyme inhibitor complex. In case of an enzyme inhibitor complex, the enzyme E and substrate S first reversibly combine to give enzyme substrate complex ES. Chemical process then occurs in second step with the rate constant K cat. The turnover number nu is the maximum number of substrate molecules converted to produce per active site of enzyme per unit time. So, it has two rate constant, E plus S with Kd goes to ES, and with K cat it goes to E plus the product. The Michaelis Menten equation. This equation connects the rate of reaction V with the substrate concentration S. The maximum rate constant V max is observed when substrate concentration is higher than enzyme. It is given by Michaelis Menten equation as nu equal to V max into S divided by Km plus S, where Km is a Michaelis constant, which is the substrate concentration at half the velocity, that is V max upon 2. Free energy of small molecule protein interaction. From chemical point of view, the important property for any small molecule protein interaction is the free energy of interaction, delta G, which is related to association constant and controls the rate of reaction. Delta G is generally given as delta G equal to delta H minus T delta S, where delta H is change in enthalpy and delta S change in entropy at any temperature T. Delta G, the free energy change at NTP, normal temperature and pressure. At equilibrium, delta G is equal to 0 and G is given as equal to KT log of Kd upon C0, where C0 is the ratio of activities of each chemical species in standard state. Theoretical computation of delta G is very important because it can tell us what small molecule or ligand can bind better than other. So, the quantity of interest is delta G equal to Gc, that is the complexed value of G, minus Gu, the uncomplexed value of G. Usually, we partition delta G into various factors as overall loss of entropy due to association, that is due to loss in translational and rotational entropy. Delta G can be expressed as alpha times delta E electrical or electrostatic plus beta times delta E van der Waal, where delta E electrostatic is the difference in the average ligand electrostatic energy with the surrounding when bound and free, and delta E van der Waal corresponds to the difference between bound and free energy in van der Waals interaction. The values of alpha and beta can be estimated from base bits. One can also add more terms like H bonding and others. Physical basis of small molecule protein interaction. The free energy change delta G between small molecule and protein receptor is the free energy changes associated with each individual components of energy. Initial long range attraction between the drug and receptor is almost entirely due to electrostatic component. Electrostatic interaction. U electrostatic is given by 332 Qi Qj divided by epsilon r, where epsilon is the dielectric coefficient with the value 1 for gases, 4 for hydrophobic environment at the center of protein, and up to 28 for interactions occurring at the surface of protein, and finally 80 for bulk water. One can partially accommodate this extreme dielectric constant using a distance dependent dielectric constant equal to Rij, where here Rij or R abbreviated is the distance between the two charges Qi and Qj. This shows that electrostatic interaction is a very important for interaction between small molecule and protein. This initiated that it is necessary to have electrostatic complementarity between the small molecule and protein, and number of programs became available for computation of electrostatic potential around small molecules to find out the recognition or the molecules with better recognition for a particular active cavity of a protein. Nonpolar and H bonding interaction. Nonpolar interactions occur amongst nonpolar molecules, especially when these are at a short distance. For example, U non is equal to A by R12 minus B by R6, again we drop the suffixes Ij, and A by R12 is a repulsive term minus B upon R6 is a attractive term, and H bonding interactions are amongst the bonding donors and acceptors. It is a 10-12 term and given by the equation C upon R12 minus D upon R10, where A, B, C, D in all these equations are numerical constants. A and B determined from polarity, and R is the distance in this case between the proton donors and acceptors, in the first case between the non-bonded pairs of atoms. Another interesting or important contribution is the hydrophobic contribution. And again, considering its importance, number of computer softwares became available for computation hydrophobic maps around the molecule. The interaction is generally described by equation Fj equal to minus 2.3 kT, summed over all the fragments, Fi into exponential to the power minus Rij, and summation is your number of fragments, where Fi is the fragment hydrophobicity and T the temperature. Free energy of interaction between small molecules and protein. Accuracy of calculation of binding free energies depend on correct prediction of properties of small molecule and any changes in those properties arising from the complexation to the macromolecular binding site. This is a very, very important parameter for designing of a drug or a ligand which should have a better effect than the previously known ligands. So, one must select first the appropriate molecular species, then one must be able to accurately predict the value and contributions to the ligand solvation free energies. Free energy computation. Since absolute free energy computations are difficult, thermodynamic cycle perturbation approach is used to calculate relative solvation free energy delta G sol between two solutes. The method gradually converts one solute into another. We show in this picture thermodynamic cycle for solvation of ethane to methanol. Here the suffix G is for gaseous conversion and AQ for aqueous conversion. Experimental techniques. Variety of experimental techniques can be used to study the interaction of ligands, small molecules with proteins. Spectroscopic techniques, UV, fluorescence, infrared, FTIR, Raman, CD have been used. The studies are facilitated by naturally occurring chromophores in protein structures. If these are involved in interaction, their spectra gets modified and one can know the interaction. Mass spectroscopy, atomic force microscopy had been of great help. NMR and X-ray crystallography had given over 22,000 complex structures between ligand and receptor. Computer-aided modeling. It basically involves either a 3D database search for a specific target on the basis of interactions between ligand and protein or target using some scoring functions, or de novo design. These days there are several packages as Gold, Autodoc, Ludi, easily available on net for CAD, computer-aided drug designing, which can be used for ligand designing. Initially, one has to optimize several other properties such as bioavailability, metabolic half-life. These can be optimized using programs again available on net as ADMET, PASS, Mole Inspiration, and there are more. One uses quantitative structure activity relationship QSAR, and this can be studied again using packages as MOE and CBIL, SYBYL. CAD, computer-aided drug designing. This technique has become very, very popular with the pharmaceutical industry because of the very high cost and long span involved for discovering a new drug molecule. But the success of the CAD depends on the scoring functions used for the study. Another thing, if you use QSAR, then the accuracy of the descriptors and the choice of descriptors is very important. There are many successful stories of designed drugs. For example, carbonic anhydrase inhibitor dorzolamide, tyrosine kinase inhibitor imatinib, H2 receptor antagonist climetidine, selective COX-2 inhibitor of non-steroidal anti-inflammatory drug family, celecoxib, iterecoxib, HIV entry inhibitor, enfuvirtide, and there are many others. Let us now see some specific examples of interaction of small molecules with proteins. The first example I want to give is HIV-1 protease. HIV is a lentivirus. It has two major species, HIV-1 and HIV-2. HIV-2 is not so important or carries low risk of transmission of HIV than HIV-1, which causes AIDS. And HIV-1 protease is one of the best known aspartic protease and the attractive target for design of drugs active against AIDS. So to design of HIV-1 protease had been one of the greatest challenge for the design of drug. We show in this picture structure of HIV-1 protease. It has a C2 symmetric homodimer consisting of 299 amino acid monomers. One is blue, one is green here. Each monomer contributes one aspartic acid residue, ASP25 or ASP25- shown here in red color. These are essential for catalysis. There is an extended beta sheet region on the monomer known as flap region, shown here by purple color, which constitutes the substrate binding site. We show here this picture with the PDB ID 1KNG, where the flap is purple, the red are ASP. HIV-1 protease. The protease has a sequence ASP THR GLI, which is conserved amongst mammalian aspartic enzymes. These lie at the bottom of hydrophobic cavity. Water molecule plays an important role in opening and closing of the flap region seen earlier, as well as increasing the affinity of enzyme with the substrate. The aspartyl residues are involved in hydrolysis of peptide bond. The preference cleavage site is N-terminal side of proline between Phe-pro or tyrosine proline. Design of HIV-1 protease inhibitors. Just after the discovery of HIV, number of experimental and theoretical techniques were applied for the design of a suitable inhibitor because of the great clinical significance and human problem worldwide. AIDS was the very disease which was so threatening to the whole world population. HIV-1 protease inhibitors, a very highly selective inhibitor saquinavir was developed just within 10 years of the discovery of HIV-1 protease and approved by FDA. This is one of the very fastest discovery of a drug in the case of HIV treatment. This was followed by discovery of more such drugs, ritonavir, indinavir, etc. We show here the picture of HIV-1 protease with the inhibitors saquinavir with the PDB-ID of the structure is 2NMZ and the structure is by Tai et al., which has come pretty recently, 2007 only. We show here the chemical structures of HIV-1 protease inhibitors saquinavir, ritonavir, indinavir, and you can notice a typical pattern in this structure. I won't go into the details. How does saquinavir bind to HIV-1 protease? This was studied by double mutants G48V and L90M. Then structure was solved and studied, and they found that because of valine 48 inhibitor moves away from the protease, resulting into reduced activity. Van der Waals interaction of M90 with the main chain atoms at the active site reduce the volume of and structural flexibility of S1F1- substrate binding pocket. This confirms involvement of G48 and L90 in saquinavir binding and helps us to design more ligands. Design of NSAIDs as COX-2 selective inhibitors. Cyclooxygenase COX are enzymes involved in conversion of arachidonic acid into prostaglandin H2, PGH2. It has two forms, COX-1 and COX-2. COX-1 is an enzyme responsible for synthesis of prostaglandins and thromboxanes in gastrointestinal tract and blood platelets. COX-2 plays a major role in prostaglandin synthesis in inflammatory cells and central nervous function and is a target for design of non-steroidal anti-inflammatory drugs. For your information, these are the drugs used as painkillers, and practically all of us take it so often, like aspirin, any analgesic drugs, and so on. So this is not that they are given in a very specific disease, but these are daily used drugs. But normal problem is that if the drug inhibits both COX-1 and COX-2, then we get side effects because of the disturbance in gastrointestinal tract. But if the drug or the ligand would bind to only COX-2, then we would not have side effects. So our objective is to design a molecule which would be selective binding to COX-2 only. So let us see the structure of COX-2. Consists of a short N-terminal epidermal growth factor domain, which is shown in red color, helical membrane binding domain, which is residues 111 to 122, green region, C-terminal catalytic domain shown by blue, the drug molecule shown in yellow, and Heim group in red. The COX active site consists of a long narrow channel extending from the outer surface of the membrane through a narrow entrance restricted by H-bonding network between arginine 120, GLU 524 and tyrosine 355. Serine 530 in the upper portion of the channel and tyrosine 385 is at the apex. A long and sleek molecule as flurbiprofen, we take it very often, can bind to COX-1 active cavity. We show picture of flurbiprofen with COX-1. It sits nicely in the narrow cavity and interacts. PDB ID is 1CQE. The catalytic center of isoenzyme COX-2 is almost identical to that of COX-1. So it's very difficult task to make a molecule which will bind only to COX-2 and not to COX-1. There are small differences that isoleucine 523 and histidine 513 in COX-1 are replaced by valine 523 and arginine 513 respectively in COX-2. Residues which interact with NSAIDs, namely tyrosine 385 at the apex of channel, serine 530 in the upper portion, arginine 120 in the midway, are same. COX-1 and COX-2 active cavities are shown in this picture. The only difference between COX-2 and COX-1 channel is the COX-2 channel is 17% larger volume than COX-1. Volume 316 and COX-2 channel is 394 angstrom cube. The lengths of the channels are similar, 25 angstrom, width around 8 angstrom. Hence, the key for developing COX-2 selective drugs is to make use of its larger active cavity in COX-2. I wish to show you few more differences in the active cavities of COX-2 and COX-1. COX-2 channel consists of three distinct regions. Region 1 extending from hydrophobic pocket around tyrosine 385, ending at GLU 524. Region 2 extending from serine 530 to arginine 120. And region 3, which is more present in COX-2 only, is above histidine 90 and arginine 530. And one can make use of design a small molecule which will fit only to COX-2 and not COX-1. This has been done in design of COX-2 selective inhibitors. We show here 4 inhibitors: DuPont 697, Selective, Trophacoxib, Valdecoxib, and all of these interact better with COX-2 and cannot enter COX-1 cavity. I would like you a very good example, a specific inhibitor for cyclooxygenase 2, that is COX-2. Selective, which is used as a COX-2 selective inhibitor, has a very interesting interaction with COX-2. Its bulky sulfonamide group of Selective and Trophacoxib prevents the molecules from entering the COX-1 channel, cyclooxygenase 1 channel, because that COX-1 channel is a narrow and these molecules are bulkier. 4-methylsulfonyl attached to an unsaturated, usually a 5-member ring with the vicinal lipophilic group is required for the specificity of this interaction. The SH2CH3 group can be replaced by SO2 NH2, whereas the lipophilic packet is occupied by an optimally substituted phenyl ring or a bulky alkaloid substituent. I wish to give you another example from immunology, the interaction of viral peptide with MSC class 1 protein. MSC class 1 protein binds to peptides generated by degradation of cytosolic protein by proteasomes. It is then inserted in plasma membrane and bound to the external part of the cell. When a cell expresses foreign protein, such as after viral infection, a fraction of the class 1 MSC will display these peptides on the cell surface. Consequently, CTLs, cytosolic T cells, specific for MSC peptide complex, will recognize it and kill the presenting cell. Let us see the structure of MSC class 1 protein. It has 2 polypeptide chains, heavy chain about 360 residues and lighter chain about 99 residues. The lighter chain is called B2M microglobin. The extracellular portion is divided in 3 domains: alpha 1, alpha 2, alpha 3, about 90 amino acids long. The carboxylic terminal of alpha 3 also shows sequence and structure similar to immunoglobulin. The structure of MSC class 1. The structure is divided into 2 globular regions, one from alpha 3 and other from beta M. Alpha 3 is attached to membrane surface and region closest to the membrane surface. The domains are packed together through 4-stranded beta M sheet. The helical regions are above the floor and almost parallel, separated by 18 angstrom. The large cerevisiae that face the solution is thus formed, with the floor forming the bottom and helices the side, which is an antigen binding site. We show in this picture HLA class 1 protein structure. Here the beta sheet region and surrounded by 2 alpha helices, blue and green, can be seen. The peptide bind with their N and C terminal, buried in peptide binding site, with their central region bulging from the cleft. The peptide binding to MSC class 1 is shown schematically in this picture. The simplified model of nanopeptide with 1 MSC and 6 pockets are labeled from A to F. The main anchoring sites are from 2 to. I wish to give you another example. This is the interaction of membrane binding protein bacteriorhodopsin with retinol. The bacteriorhodopsin is an integral membrane protein found in 2-dimensional crystalline patches known as purple membrane, which occupy 50% of the surface area of archival cells. The repeating unit of hexagonal lattice is 3 identical protein chains, each rotated by 120 degrees. Each chain has 7 transmembrane helices and contains 1 molecule of retinol buried deep within. We show in the picture structure of bacteriorhodopsin with retinol in purple color, whereas here we show a trimer where the 3 units, 3 protein chain are arranged in a symmetry. How does retinol act? Retinol binding protein bacteriorhodopsin, the light absorbing pigment retinol. When it absorbs light, there is a conformational change called photoisomerization. Now, this activates the proton pump in the retina. So, retinol is covalently linked to lysine 216 in chromophore by shift base action. After photoisomerization of the retinol molecule, ASP85 becomes proton acceptor of the donor proton from the retinal molecule. This releases proton from the holding site into the extracellular side, EC, of the membrane. Reprotonation of the retinal molecule by AsP96 restores its original isomerized form, and this results in second proton being released to extracellular side. AsP185 releases its portion in holding site. So students, what have we learnt from this today's presentation? We learned first the thermodynamic and physical basis for small molecule interaction with the protein. We went through the experimental and theoretical techniques which can be used to study the interaction of small molecules with proteins. Then we discussed few selected examples between small molecule and protein interactions. First, we saw the interaction of HIV-1 protease with inhibitor. This is used for the design of anti-AIDS drugs. Next, we saw design of NSAIDs, that is non-steroidal anti-inflammatory drugs, which should be selective inhibitors of COX-2. They can remove pain without giving any side effect, and all of us take tons of painkillers. So we want to have painkillers which should not have gastrointestinal disorders. Interaction of MSC class 1 proteins with viral peptides was seen, and this is a typical example in the immunology, how we respond to a specific peptide and specific immune response is triggered. Then we saw light-driven proton probe, how conformational changes in retinal and its interaction with bacterial rhodopsin takes place. Thank you very much.